Mitosis is the process of nuclear division in eukaryotic cells. During interphase, the nucleus is enclosed in the nuclear envelope. A single centrosome duplicates, forming two centrosomes. Centrosomes are regions that organize the microtubules of the spindle. In animal cells, each centrosome contains two centrioles. The cell grows during G1 and G2 phase, and chromosomes are duplicated during S phase, but they can't be seen individually because they are uncondensed. In prophase, the chromatin fibers coil tightly and condense into individual chromosomes, and the nucleoli disappear. Each duplicated chromosome appears as two identical chromatids, connected by a centromere. The mitotic spindle begins to form. It is composed of centrosomes at the ends and microtubules that extend from them. The radial arrays of short microtubules that extend from the centrosomes are called asters. The microtubules lengthen, and the centrosomes move away from each other. In prometaphase, the nuclear envelope fragments, allowing the microtubules to extend to the nuclear area from each centrosome. Chromosomes condense even more, as each of the two sister chromatids now have a kinetochore, which is a specialized protein structure at the centromere that is assembled on specific sections of DNA at each centromere. Some of the spindle microtubules attach to the kinetochores, or kinetochore microtubules. When they attach, the chromosome begins to move toward the pole. But the microtubules from the opposite pole also attach to the kinetochore on the other chromatid, leading to a tug-of-war movement and jerking of the chromosomes back and forth. Non-kinetochore microtubules interact with those from the opposite pole of the spindle. Metaphase is the longest stage of mitosis. During metaphase, the centrosomes are now at opposite poles of the cell. The chromosomes all line up at the metaphase plate, which is an imaginary plate that is equidistant between the spindle's two poles. In each chromosome, the kinetochores of the sister chromatids are attached to kinetochore microtubules coming from opposite poles. Anaphase is the shortest stage of mitosis and lasts only a few minutes. Anaphase begins when the cohesin proteins are cleaved, causing the two sister chromatids to suddenly separate, and the two sister chromatids become individual chromosomes. The two chromosomes move to opposite ends of the cell as their kinetochore microtubules shorten, and they are pulled by their centromeres first, because the microtubules are attached to the centromere. The cell elongates as the non-kinetochore microtubules lengthen, and at the end of anaphase, the two ends of the cell have an equal number of chromosomes, a complete set. After anaphase comes telophase. In this phase, two daughter nuclei form within the single cell. This starts by having the nuclear envelope starting to appear, forming from fragments of the parent cell's nuclear envelope, along with other portions of the endomembrane system. The nucleoli then start to appear, and the chromosomes become less condensed. The remaining spindle microtubules are then depolymerized. At this point, there are two nuclei that are genetically identical to themselves and the parent nucleus. The cytoplasm is split in late telophase. The last stage of mitosis is cytokinesis. In cytokinesis, a cleavage furrow forms in animal cells. The cleavage furrow is a shallow groove in the cell surface near the old metaphase plate. The cleavage furrow then pinches the cell in two. The end product of cytokinesis is two daughter cells with a diploid number of chromosomes. In plant cells, the cell wall enlarges until its surrounding membrane fuses with the plasma membrane along the perimeter of the cell. Mitosis is now complete.